This report presents an assessment of the market for commercially available biosynthetic or produced outside the body red blood cells RBCs. Note that this product offering is distinct from existing blood fractionation and storage technologies and from cellular agriculture approaches focused on producing animal-derived blood products. This analysis will focus on companies that produce lab-grown RBCs either from stem cells or progenitor cells and crucially not from human donors. Despite being in a nascent commercial phase, this space is quickly gaining attention through clinical trials, research and development investments, and strategic corporate partnerships. Key market drivers include donor supply limitations, especially for rare blood types, as well as an aging global population and the need for universal or rare blood type replacements. Other growth vectors include military and disaster medicine applications, as well as growing interest in engineered RBCs as therapeutic carriers, such as for enzymes, antigens, and other biomolecules. However, several challenges remain. These include high production cost, typically $5,000 to $10,000 per transfusion unit, as well as the need for GMP compliance and navigating complex regulatory pathways. Most current processes lack automated manufacturing capabilities and scalability, with many processes taking 3 weeks or more per batch. Several companies are pursuing this opportunity. Rubius Therapeutics, a US-based company, has developed a proprietary platform for red cell therapeutics, which are enucleated RBCs derived from CD3 for progenitor cells. These cells can be genetically engineered to carry therapeutic payloads. Initially focused on enzyme replacement and rare disease therapy, Rubius later shifted focus to immuno-oncology using red cells as delivery vehicles for checkpoint agonists and tumor antigens. Next, we have EryPharm, a French company and subsidiary of 𝗕𝗟𝗢𝗢𝗓-𝗔𝗬𝗘𝗧𝗓, or EFS. EryPharm is one of the first groups to clinically evaluate lab-grown RBCs from hematopoietic stem cells. Their technology aims to provide viable alternatives for rare blood types and immunologically complex patients. EryPharm successfully completed a first-in-human clinical trial between 2011 and 2016, demonstrating the safety of transfused cultured RBCs. Their platform uses clinical-grade, GMP-compliant processes and is backed by substantial French and EU public funding. However, production remains slow and expensive, about 3 weeks per unit and more than €8,000 per unit, limiting immediate scale-up potential. Finally, we have Megakaryon Corporation based in Japan. Megakaryon is focused on iPSC-derived RBC and platelet production using a proprietary platform that creates immortalized progenitor cell lines. They collaborate with leading Japanese institutions and industrial partners to scale up production systems. RBC production is currently in the preclinical stage, while platelet production is closer to clinical trials. Key strengths include scalable stem cell technology and strong partnerships with industrial and academic players. However, the complexity of regulatory approvals and the need for further clinical validation represent key challenges. In conclusion, early strategic entry into this space via partnerships or licensing could provide a significant competitive advantage in a future where synthetic blood becomes a critical healthcare infrastructure component.